This video is about the immune system, the immunity. We saw everything with the lymphocytes and antibodies. You see the two common terms, that is antigen and antibody. The antigen is something against which the antibody is produced. The antigen comes from outside. It is located on the surface of the bacteria, on the other virus, on the fungi, on the anything which can generate the antibodies can be called as antigen. So here you see that this is if an antigen molecule, it has this specific angle to which the antibody can attach. Mostly antigens are, it can be small as a small oligosaccharide, it can be small chain of the protein, amino acid. So antigens are present against which the antibodies are produced. The structure of the antibody, if you see, it has 4 polypeptide chains. The 2 chains are light and 2 are heavy. It is typical Y-shaped structure of this antibody. And this site is the place where the antigen will bind, the antibody will be binding to the antigen. So this is variable region, but this is the constant region. तो वाई जितने तरह के एंटीजन होंगे उतनी तरह की एंटीबॉडीज बन सकती हैं। इवन एंटीबॉडीज के खिलाफ भी एंटीबॉडीज बन सकती है। इवन एंटीबॉडीज के खिलाफ भी एंटीबॉडीज बन सकती है। वी सी लेटर ऑन। So if you see this antibodies are present in the plasma. In this, the plasma has globulin protein. You see, the globulin are 3 fractions: the alpha, beta, and gamma fraction. In gamma fraction of the globulin, the immunoglobulins are present. Immunoglobulins are these antibodies are present. So whenever something external comes, so the antibodies are produced. So here we are seeing this in this picture, that what happens if the antigen antibody reaction takes place. Here this you see, this is the bacteria, for example any bacteria. On this surface these are antigens, right? And it has entered into the blood. So against this antibodies have been produced. These antibodies can bind antigen. So if this was such type of antibody like this, which has 5 arms, so it can bind 4 or 5 bacteria at one hand, and so it will cause it agglutination. These bacteria can no longer move, they can no longer enter into us, and further what will happen, the macrophage or neutrophil will come and will eat this bacteria which is not now being able to move. So this is one function of the antibodies, that they cause agglutination, right? Agglutination. Here we are also seeing the antigen antibody reaction leading to agglutination. Here these are the antibodies which have bound the all cells which are having the antigens. If you see the types of the antibodies and type of the lymphocytes, this is one thing I would say. This is the neutrophils, eosinophils, basophils, macrophages. They are the type of the immunity that they are not specific for a particular type of the bacteria. Any type of bacteria comes, neutrophil can eat. Any type of the foreign body state, the macrophage can eat. So these cells which are neutrophils, basophils, dendritic cells, macrophages, mast cells, these and natural killer cells, they give the immunity which is not specific for a particular type of the bacteria, a particular type of the virus. So it is non-specific immunity. But when we come to a specific immunity, which is being adaptive and which is against a particular cell, particular bacteria, then that is we call it with the help of the lymphocytes. Here we are seeing there are 2 types of lymphocyte. One are called T lymphocyte and one is called B lymphocyte. The T lymphocyte are the cells. T lymphocyte get their schooling in the thymus, and B lymphocyte get the schooling in the bone marrow. B for bone marrow, T for the thymus. So what is that T and B lymphocytes do? We see that now. They are located in the lymph nodes. They are located in the lymph. They are located in the blood, and they are located wherever there is the chances of infection to come, like as in the mouth, the air enters, in the food enters. So there are the lymph nodes. In these lymph nodes are the lymphocytes. Those lymphocytes which have got their origin from the bone marrow, then in the bone marrow, the B lymphocytes have understood that what is their own antigens, and against those antigens they have not to produce antibodies. If there is such type of the B lymphocyte which is producing the antibody against the self antigen, is being destroyed itself in the bone marrow, it's not allowed to move into the blood. Similarly, in the T lymphocyte, in the thymus, they get the training so that um, don't release those T lymphocytes which have the immunity against, which are toxic against the self antigens. So these are difference between the T and the B lymphocyte. Now, how they act? What are the types of T cells? What are type of B? What B cells do? Here we're seeing this picture. See, if say this is a macrophage and it has eaten the bacteria, and it will show that I have eaten this. मतलब ये बैक्टीरिया बताएगा कि मैंने क्या खाया, ये मैक्रोफेज बताएगा कि मैंने क्या खाया. In the form of, it will represent the antigens on its surface. It did the phagy of this infected cell and it got this antigens, and now it is showing those antigens on its surface, like as in the antigen MHC complex. These antigens are now presented to the T cells. There are four types of T cells. One is the helper T cells, which are going to help the B lymphocyte to produce the antibodies. There are memory T cells which are going to remember for 4-5 years that this type of antigen was in present, and whenever again the same antigen will appear, same type of infection will appear, this will has a memory, and this can again develop into the T cells which can destroy, that can change to killer T cells and can change to the helper T cells. So these are the memory T cells. The third type of the cells are killer T cells. The memory T cells we saw, which have the memory. Then helper T cells we saw with their helping the B lymphocytes to produce the antibody. These are killer T cells which can directly produce such things which can kill this infected cell or the cancer, and so this is the type of immunity which is by T lymphocytes. The B lymphocytes, when with the help of the helper T cells, they will convert into the plasma cells and they will produce the antibodies. So there are three antigen presenting cells we saw. One we saw the macrophage, the other is dendritic cells, and some of the B cells also act as the antigen presenting cells. Means here it is saying that after eating, after phagocytosis, they are presenting their antigen, the antigen on the surface, right? So that the others can know which type of the antibody they have to produce. This is a typical structure of the antibody which has been produced from B cells, from the plasma cell. There are 5 types of the antibodies: M, mega, G, gamma, A, alpha, E, epsilon, D, delta. So these various type of antibodies are produced by the plasma cells. The IgE is mainly responsible for the allergic reaction. This will stimulate the eosinophils. IgM is produced initially. It will cause coagulation. Then later on, IgG will be produced. IgA is the secretory immunity. It is produced by the mucosal cells. So and D is again the immunity which acts on the B cell receptor. So I want this way the cells will be the cells will provide the immunity. The cytotoxic, the T cell immunity is called cytotoxic immunity, and the B cell give the humoral immunity. The B cell produce the antibodies, and C cells are going to directly kill the cell. So if when the grafting is done, then also if the graft doesn't match, then these T cells will come and will destroy the that type of the